So now I want to talk about what happens when your sodium current, right? We have our sodium ions that are rushing down, that are propagating down the axon, right, heading towards the axon terminal. What happens when they reach the end of the axon, when they get to the end of the neuron? Because at that point, there has to be the coupling of the arrival of that current, right? The arrival of those sodium ions has to be coupled to the release of a chemical messenger, a neurotransmitter that is going to exit this first cell and influence the function of the next cell, perhaps stimulating an action potential in the next cell. Now, this is a very complicated drawing, and I promise we'll go over it. So here is our axon, right? You can see the sodium coming down that axon terminal. That's our action potential heading down towards the end of the axon. You can see we have our voltage-gated sodium channels right up there, right, on the axon. So as the sodium comes down, it's a positive ion, right? It's going to be depolarizing, the cell, from -65 to the threshold of -55, and those voltage-gated sodium channels are going to shift and open and allow for the entry of more sodium. So that current is going to be propagated, right, all the way to the end of the axon. Now, what I want you to notice is, at the end of the axon, there's a lot more stuff, right? We have a lot more structures that are here. Remember, all of these are built up, based upon instructions in your genes, and they are sent to particular locations inside a neuron. So instead of the sodium channels, the voltage-gated sodium channels, when you reach the axon terminal, you're going to encounter another kind of voltage-gated channel. It's another channel that is going to change its shape, right, depending upon the charge difference between the inside and the out. And it turns out that in order to open these channels, right, these voltage-gated channels, you have to reach a particular threshold voltage. At the resting potential, they're going to be closed the same way the voltage-gated sodium and the voltage-gated potassium channels are closed. So when sodium comes rushing down, right, it's depolarizing. Remember, it's going from -65 to -55. When it arrives at the axon terminal and depolarizes to threshold, it turns out it's about the same as the threshold that we've seen for the sodium and potassium channels, about -55 millivolts, right? These voltage-gated calcium channels will open. Calcium will, enter the cell, and it will come in upon the arrival, of the sodium current. I want you to hold that thought, because we'll talk about this in a little bit more detail, about what's going to happen at the synapse. Well, let's describe some of the structural features that we see here. So at the axon terminal, we no longer have voltage-gated sodium channels. We have voltage-gated calcium channels. Okay? We also have clearly a number of these spherical, what are known as vesicles. These are membrane-bound, so they're composed of phospholipid bilayer, right? They're these little kind of, spherical phospholipid bilayer bound sacs, right? And they are, embedded with proteins, right? And these proteins are a particular type of machine, right? These are, let's see if you can figure this out. These are proteins embedded in the vesicles that pull in using ATP, right? Using energy, right? Pull in neurotransmitters and fill these vesicles with chemical neurotransmitter, okay? So a protein structure, right, that uses energy to grab onto something on one side of a membrane and fight a force to pull it inside, this vesicle, is known as a transporter. This is called the vesicular transporter because it's on the vesicle. The vesicular transporter grabs on a neurotransmitter, fights a force to concentrate it inside the vesicle. Think about what force is requiring the use of chemical energy here. If it's fighting to concentrate neurotransmitter inside that vesicle, it's fighting the concentration gradient force, right? So it's actually storing energy here. Think about this. As you pack more and more and more neurotransmitter inside the vesicle, if you were to open a gap or a hole in that vesicle, the concentration gradient force would provoke, rapid outflow, right, of the chemical from that enclosed, tightly packed, highly concentrated space. Okay. I want you to see there's some other structures that are located here. There are a whole bunch of proteins here. I just picked a number of different shapes, right? They're actually sort of docking this vesicle, which is now pretty much filled with neurotransmitter here, right, to the membrane, right near the membrane of the axon terminal, okay? I'll show you, this one actually shows, a continuous membrane, and we'll talk about this in another video lecture. This shows neurotransmitter entering what's called the synaptic cleft. There's a gap between the axon terminal, right, of one neuron and the what's called postsynaptic membrane, after the synapse membrane of the next. This is actually also called the presynaptic membrane. Pre is before. It's before the synapse or space, and post is after, right? So think about how you would identify these different components, right, of a typical synapse. If you have the axon terminal or presynaptic membrane, you're going to look for the presence of voltage-gated calcium channels. You're going to look for the presence of vesicles that are coated with vesicular transporters. You're going to look for the presence of neurotransmitters that are being packed inside of those vesicles. You're going to see vesicles docked, these complex protein structures, right, to the membrane, of the axon terminal presynaptic membrane. You're also going to see some, under certain conditions, right, releasing their neurotransmitter into the cleft. The postsynaptic membrane can be identified by the presence of rather large protein structures, okay? These are actually the structures that are going to receive the chemical messages that are being released by the presynaptic neuron, right, the axon terminal. So these are called receptors. And I want you to note, there's a number of different kinds of receptors. There's at least 2 distinct shapes that I have written up here. This is going to become very significant in terms of what these receptors can do to the postsynaptic cell.